Hi, today we'll talk about immunologic assay. So antigen antibody reactivity and cytokines, drugs and cells can be detected in vitro and in vivo, can be detected up till 2 nanograms or picogram range. The union of antigen with antibody is very sensitive, specific and firm, but reversible. Binding occurs in seconds but is not visible until something called lattice forms, which occurs more slowly. The composition of lattice depends on the ratio of the antigen to the antibody. The affinity is the measures the binding energy between an antibody and univalent epitope, while avidity is the total binding energy between an antibody and the multivalent antigen. The specificity of an epitope can be changed by changing the position of atoms, double bond, structure conformation, or the composition of its amino acids or sugars. From this figure, you can see that the size of antigen antibody complexes is determined by the ratio of antigen to antibody. In case of larger complex, in case of antibody excess or at equivalent, these larger complexes are phagocytized. But in cases of a smaller complexes like in case in antigen excess, represented here by the orange circles, the antigen escape and lodge in blood vessels and behind the renal basement membrane, causing vasculitis and glomerulonephritis. Protection test. So protection tests are used to determine the potency of a vaccine. The active. Following immunization with the vaccine that is being tested, groups of animals are challenged with increasing numbers of microorganisms. The lowest number of microorganisms lethal for 50% of the animal, that's called LD50 or lethal dose, that's responsible for which is lethal to 50% of the animal, is determined and compared to LD50 in non-vaccinated animal in order to measure the protective effect or power of the vaccine. Passive. It's a graded amount of serum from immunized individuals are transferred to normal animals, which are then challenged with the infectious agent. The highest dilution of serum effective at protecting 50% of the animal, that's ED50 or effective dose 50, is determined as measure of the efficacy of the vaccine. So here you can see that you either produce the immune in the organism actively or passively. Agglutination tests. These tests are used to detect antibody union with large particulate antigen. These are rapid slide identification of bacteria. So bacteria from patient's culture plus a battery of specific antibacterial antisera, you must note that which antisera causes the agglutination. It's a semi-quantitative diagnostic test for bacterial disease. Suspect bacterium killed um, is added to dilution of patient's serum. So the highest dilution that results in a visible agglutination is called the titer. A fourfold increase in titer is necessary for diagnosis. Slide agglutination is used in blood grouping to determine whether the donor cells or serum possess antigen or antibodies that are reactive with recipient serum or cells. There is a major cross-match when the donor cells plus the recipient serum to determine whether the anti-RBC antibodies are present in the recipient serum. So that occur rapid clumping of donor cells will occur in vivo if the anti-donor RBCs are present. While minor cross-match, it is when the donor's serum added to the recipient cells and there is agglutination of the recipient cells occur if anti-RBC antibody are present in the donor serum. So the transfusion reaction under minor cross-match is much less severe than the one associated with a major cross-match, because the amount of antibody transfused in the donor serum is minimal relative to the number of RBCs in the recipient. Comes test as a test of agglutination test. So Comes test is either direct or indirect. In indirect tests, there is a detection of the weak or non-agglutinating anti-Rh antibody by adding anti-human immunoglobulins directly to the infant's RBCs, to which the Rh antibody has been attached in utero, and observe and notice any agglutination. In this figure, you can see that the infant RBCs already have the Rh antibody bind on its surface. When we add the anti-human immunoglobulin to this sample of blood, and the positive test, if there is Rh antibody, the positive test will result as agglutination, because anti-human antibodies forms links between RBCs by binding to the human antibodies on the RBCs, and this is the direct test. While in COMPS indirect test, you measure the same weak antibody in the maternal circulation by adding the mother's serum into the RBCs in vitro. The addition of anti-human immunoglobulin will result in agglutination of the RBCs, and this is a positive test. So in this figure, you collect the recipient's serum, suppose that it have the Rh antibody, you incubate with red blood cells. If there is Rh antibody, theoretically it will bind on the surface of the RBCs. When adding the anti-human IgG, which is a COMS antibody, is added to the solution, the positive test will be shown as agglutination of red blood cells occur, because the human IgG are attached to the antibody that bound to the RBCs. So we'll talk about continuing the agglutination test about the IgM complement fixing antibody. These used to agglutinate RBCs at temperature below 37 degrees and can be detected by incubation at lower temperature. These cold agglutinins are frequently autoimmune in nature and occur commonly in patients with primary atypical pneumonia caused by the Mycoplasma pneumonia. Another test is diagnostic test for a mix of viruses like influenza and mumps. These involve the neutralization of their ability to agglutinate RBCs by infected patient serum. The patient serum titer is determined by the highest dilution that block the agglutination. Precipitation reactions. It is used to detect soluble proteins, polysaccharides, and antigen antibody complexes. Either antigen or antibody in serum can be measured quantitatively with analytical precision. The increasing amounts of antigen are added in a separate tubes containing a constant amount of patient serum. The resulting precipitate in each tube with the increasing concentration of the antigen is washed and analyzed, and the precipitated antibody is plotted as a function of antigen added. So probably three zones results from the antibody excess, equivalence, and antigen excess. Immunoelectrophoresis. It is used as a type of precipitation reactions as well. It's used to identify specific antigen in a mixture and in immunological disorder. Components of an antigen mixture are separated in agar, first by migration in electric field, followed by their diffusion and subsequent precipitation with a specific antibody diffusing from an overhead trough. So you can see in this simplified figure, you can see that antigen and antibody are located in the agarose on different ends. The antibody near the anode and the antigen is near the cathode, and there is a migration in the agarose for both the antigen and antibody until they meet. So this is identified by a precipitation line, and you can see and you can calculate the concentrations of the antigen and antibody, and to see how much antigen is more or antibody or more by the location of the precipitation line. Third type is radioimmunoassay, RIA. It is based on the displacement of known radiolabeled antigen from an antigen antibody complex by unknown unlabeled antigen, like a hormone, in a patient's body fluid. The extent of loss of the labeled antigen from these complex can be measured and is a function of the concentration of the unknown antigen in the patient's fluid. Can measure sensitivity is less than 1 nanogram. So you can see from this figure you have a test tube that you add the radiolabeled drug and the antibody, and there will be a precipitate protein, and in the supernatant you can see no radioactivity in the supernatant. While here, when you add the radiolabeled drug with the unlabeled drug with the antibody, and you do a precipitation reaction, you can see the amount of radioactivity in the supernatant. The fourth type is the radioimmunosorbent test, RIST. It is used to measure the total non-specific IgE in allergic patient's serum. The patient's serum is added to a rapid anti-human IgE, which has been absorbed onto the particulate agarose beads, and the complex is washed. The iodine 125 labeled rabbit anti-human IgE is added afterward and again washed, and the amount of radioactivity is quantified, which reflects the total concentration of IgE in the patient's serum. The fifth test is radio allergy sorbent test, RAST, and it's used to measure the IgE in a patient's serum specific for a given allergen. The suspected allergen is coupled to an insoluble matrix like above, like the agarose particles, and the patient's serum is added and washed. The iodine as well, 125 labeled rabbit anti-human IgE is added, and the complex again is washed. Why we need to wash after each time we add? To remove the excess and bound. And the bound radioactivity is quantified, reflecting the serum level of IgE specific for that allergen. So basically, in these two techniques, the labeled rabbit anti-human IgE will bind to IgE if it's present. So if it's not present, it will be washed away. So if it's present, so it will release radioactivity, and we can quantify this radioactivity to know how much IgE is, how much antigen is there, and IgE actually. So you can see here a simplified figure for RIST, which measures the total serum IgE level. So you have a solid phase that have anti-human IgE, and then you incubate with human IgE. You can see that there is a capturing of IgE in patient serum. Then you add the detected by the iodine 125 labeled anti-human IgE. The amount of radioactivity recorded reflected the amount of IgE, and this is a measure, as we said, RIST for the total non-specific IgE. While the other case in the RAST, it measures the allergen-specific IgE. In other words, it used to measure the specific IgE. So you have a disk coated with the specific allergen, and you do actually the same process. You add the specific IgE bound. You incubate with the serum from the patients, and then you add the iodine 125 labeled anti-human IgE, and the amount of the recorded radioactivity will reflect the amount of the antigen present in patient's serum. The next test for precipitation reaction is the enzyme-linked immunosorbent assay, ELISA. It's a very sensitive practical assay useful in detecting either antigen or antibody in low concentration in a patient's body fluids. It's used to measure low up till nanograms concentration of antigen. Dilution of the fluids containing antigen are added to its antibody, which is adsorbed on a plastic wells. The resulting complex is washed Of course, we said before that washing is removing the excess unbound. And an enzyme conjugated antibody specific for a different epitope on the test engine is added. After washing, the enzyme substrate is added and the color reaction, which actually the readout, the color reaction is measured using a spectrophotometer, which correspond in the last test the radioactivity. And the color reaction is measured using spectrophotometer. The titer is recorded as the highest dilution of antigen, giving a color above the background. So specific antibodies in low concentration in patient serums, like in case of HIV infection, can also be measured by adding dilutions of the test antibody to its antigen adsorbed onto plastic wells. Then the complex is washed and enzyme conjugated anti-isotype antibody is added, and after washing, same procedure, the enzyme substrate is added and the color is measured and the titer is determined as above. So you can see here from the figure, a represented figure for the steps of the ELISA. Generally, you have four different types of ELISA. You have the direct ELISA, where the antigen is adsorbed on the plastic well surface, then a primary antibody, then you incubate with the serum, and then as a substrate, and then a signal will arise that reflect the amount of antigen. In another type, you have an indirect ELISA, where another antibody is introduced, is added, that is anti-human antibody, and the same procedure is occurring. In case of sandwich ELISA, that's a major difference, because in the last two, the direct and the indirect, you can see that the antigen is coated on the surface, while in this case, the antibody is coated in the surface, which is specific for the antigen, and then you incubate with the antigen, then you do the conjugated antibody with the enzyme that needs the substrate for the signal. In competitive ELISA, you have the antigen coated on the surface, and then there is another antigen which is inhibitor antigen. So you need to have the process of competition and the efficiency of binding to the antigen coated on the surface, and the amount of this signal rise from the reaction of the enzyme and substrate will tell you the amount of antigen present in the sample. The complement complex C. As you know from previous lectures, that 9 major factors from C1 to C9, most of them are pro-enzyme present in normal serum and not increased by antigenic stimulation. It is effective via 3 pathways. You are familiar with the classical pathway. It's resulting in the lysis of the microbial or mammalian cells, to which IgM or a doublet of IgG1, 2, or 3 antibody has been bound to the membrane, followed by sequential fixation of the complement to the antigen-antibody complex. To summarize classical pathway starts with the effect of the antibody and the 3 molecules C1q, C1r, and C1s, termed C1QRS. This C1QRS cleaves C2 and C4. Upon cleavage, C4b and plus C2a on the cell surface form the C4b2a, which is termed C3 convertase. This C3 convertase will cleave C3 into C3b and C3a. Soon after that, the C3b binds with the same C4b2a to form the C5 convertase. This C5 convertase will cleave C5 into C5b and C5a. Together with C6, C7, C8, and C9 will form a membrane attack complex that increase the permeability of the pathogen or the bacteria, and the change in osmotic pressure leading to the cell lysis of the bacteria. And this is the classical pathway. While for the alternate pathway, it is activated by cell walls of certain gram-negative and gram-positive bacteria and viruses, yeast, and as well aggregates of IgA. This pathway is independent of antibody, which is contradicting for the about the classical pathway. It is independent of antibody as well, C1, C4, and C2. It is initiated by cell wall absorption of small amounts of C3b. So it needed to be initiated a small amount of C3b existing in normal serum. So as a summary for the alternate pathway, you can see that it is initiated from C3 and needs a takeover from small amount of C3b. Once C3b is cleaved, C3 is cleaved into C3a and C3b, this C3b together with factor B and factor D forms the C3b B. This C3b binds with another protein, it's a pyridine, and together will form the C3 convertase, which adds to the system another C3b by cleaving C3. This extra C3b will bind with the same complex, the C3 convertase, to form the C5 convertase. This C5 convertase will cleave C5 and then again will together with the C5b, C6, 7, 8, 9 will form the membrane attack complex and will lyse the bacterial cell wall. For the third pathway, it is the mannan binding lectin pathway. It follows the binding of an acute phase protein and the mannose binding lectin MBL onto a mannose residue on the cell wall of the certain bacteria, fungi, and viruses. This complex, the whole complex, the acute phase protein mannose MBL, act similar to cellular immunity and thus follows the classical pathway forming C3 and C5 convertase that results ultimately to the lysis via the MAC. So it is an adjunct to innate immunity, but it is independent of the antibody. Complement-induced chemotaxis and opsonization. Of course, you know that fragments of C3a and C5a are potent vasodilating and chemotactic adjuvants, adding cells and cytokines to the inflammatory responses. The binding of fragment of C3b to microorganisms promote their opsonization via C3b receptor on phagocytic cells. Fluorescent antibody, another test in the immunological assay, can be used in immunological assay, and this permits the visualization of either antigen or antibody in cells or tissue. In the direct technique, fluorescinated antibody to antigen is added directly to the specimen, for example the tissue, containing the antigen and visualized under UV. The more sensitive sandwich technique is used by using dye to detect antigen. Its antibody is added first to the specimen, followed by fluorescinated anti-immunoglobulin, and the specimen is visualized under the UV. So the amount of fluorescent visualized by the UV reflected the amount of antigen present. The same can occur for detecting the antibody. The antigen is added to the specimen, followed by fluorescent antibody against the antigen, and the specimen is visualized under the UV. So you can see here how they can use different fluorescence antibody with different excitation to see different colors. You can see that in this example, these are cells have been labeled fluorescently. To the green stuff is for the some of the proteins in the cytoplasm, and the orange stuff are some of proteins in the nucleus. So you can use a multiple number of and different fluorescinated antibody to detect different proteins. The last technique is the Western blot technique. It's valuable in identifying an antigen or antibody within a mixture. The component of a mixture can be separated by electrophoresis on a sodium dodecyl sulfate polyacrylamide gel and blotted onto a nitrocellulose matrix. It is labeled and a known antibody is added to locate and identify the antigen of interest. As a confirmatory test, it can be used as a confirmatory test for the AIDS. The patient's serum suspected of containing anti-AIDS antibody is added to known HIV antigen bound to nitrocellulose matrix. Binding of labeled anti-human immunoglobulin antibody to the HIV anti-HIV complex confirms exposure. And this sketch summarize how can you detect the HIV antigen. You have a solid support, which in our case, it's in the nitrocellulose matrix, and you have the antigen adsorbed on its surface. Then you get the antibody in the sample, which is assuming to be anti-AIDS. Then you do anti-human antibody, and you have an indicator which is either a color or a fluorescent label. And this is a straightforward technique to detect a lot of antibodies and antigens. Thank you.